So now it's my pleasure then to welcome Liesbet Geris. So Liesbet is a professor at the University of Liège, and she is also active at the University of Catholic University in Leuven, involved in the Prometheus infrastructure research group or research unit. And and so she's working combining experiments and numerical modeling in order to understand the rationales that can lead to successful tissue engineering strategies. So really trying to link basic science, cellular behavior, and also potential through translational research. So Lise is doing this work in the framework also of NERC grant. And she is also, because science is not enough for her, so she's also the executive director of the VPH Institute. So
that's the pot calling the kettle black, eh? Okay, so hello everybody. I'm very happy to be here. It's slightly change of organ system, let's say, no longer heart. We're focusing on skeletal tissues, so I'll start with a very, very brief introduction into the why we are doing this and some concepts of bone. I'm really happy to be following after Alfio's talk this morning, because then I don't have to treat any of the numerics, because it has all been discussed. And actually, it's the same issues that come back, although with the heart tissues, we have slightly less problems of doing some of these couplings. So starting with the unmet medical need, it's the one where you have a shortage of organs and tissues that can be donated to patients that are in need. And when you see this graph, everybody knows this graph, you typically think of heart and lungs and liver, but in fact there's also a problem for skeletal tissues. So also bone is a problem where we don't have enough donor tissues or where we don't have enough effective tissues that we can transplant to patients. What you see here on the right is an article about a little girl, one of the patients that we're looking at in our group. She has neurofibromatosis type 1, which means that she has a genetic defect which causes very locally in her tibia, in one of her tibias, a zone of bad bone, let's say. And that is something that actually, as you see on the right-hand side, leads to this bowing and then to a fracture that does not heal. She fractured her leg when she was 4 months old. This picture was when 16 months later. Now she's 8 years old and the fracture still hasn't healed. So basically what we have to do is remove a whole big part of the bone and then be able to replace that with a living implant. So that's the patient population we're working for. Just to have a few basic notions on bone, I guess most of you have some biomedical background, so have some notions on bone. You know that bone is an intelligent material that can sense its environment and can adapt to its environment, that is capable of scarless healing in many cases, but in 5 to 10% of the cases when you have a fracture, it doesn't heal. Now, there are two approaches that we can follow in tissue engineering, and those two approaches actually mimic the bone formation that you find both in developmental stages and postnatally. That is either direct or intramembranous ossification, where you see cells aggregating inside the fibrous tissue or the mesenchymal tissues. So they become osteoblasts, they start forming bone. These become the sensor cells, so the osteocytes. When the diameter is, or the distance is getting big enough, there are hypoxia signaling, so blood vessels are attracted. And this is what you typically get in flat bones, like in the skull, where you have a cortex and some trabecular bone on the inside, with still some active bone cells on the outside. The other type of bone formation is for the long bones. It's the endochondral ossification, where you start with a template that is made out of cartilage, that then gradually through the primary ossification center goes through ossification. So you have the bone formation here, and then you have the same process happening again in the secondary ossification center, leading to the formation of a growth plate, which is the main mechanism through which we grow or we form bone postnatally through time until puberty. So those are the two strategies that you can follow also in a skeletal tissue engineering approach. Now, when we say skeletal tissue engineering, after 35 years, the field is still struggling a little bit with that. There's a lot of materials engineering going on. There's some bioreactor engineering going on. But some of the other tools of engineers, like the computer models, are still underused in this context. So one of the major challenges of tissue engineering is to take all of that biological insight that is gained every day and translate that into something that is a robust process that will give us implants that we can get through the regulatory system and that can actually work in the patient. So there we believe that also computer models can help to look at the quantification and optimization of the product, the process, and do some of the prediction, if you wish, or some of the trying to understand what happens when you implant such a living implant in vivo. Now, in order to do that, you need quite an infrastructure, and we've set up this collaborative group, which is called Prometheus, for obvious reasons, where we work from the basic biology side, so looking at growth factors. We have people working on different types of stem cells that could be used, looking at the mechanisms of action when you put all of these basic building blocks together. And then when we have something that works in a mouse, we need to be able to find a way to translate this to a sheep and then to a human. So we have process engineering, looking at bioreactors, upscaling. We have the large animal experiments that we do, and then we have our patient population already selected. All along, we have the enabling technologies, and the modeling part is the thing that I want to discuss today. Just to give you an idea of the amount of people that you need, these are my team members, and these are the other people also in Prometheus taken at our retreats and our group activities, because you need to bring all these people together, not only in a science way. Now, when we want to make a model, we don't want to just make a model, we want to answer a question or try and help answer a question. So everything we do is in a kind of integrative way. There's always a question where there is some observations or a biological question, a hypothesis that is somehow formulated, or just a vague question, where there's maybe an experiment that leads to data, and then we can make a model to further look at the data or to have some insights into what we should test actually in the experiment, and then that feeds back into the experimental part. Another loop, this time for the modeling alone, is the life cycle of a model. So you start with a question, you try to pick your modeling strategy, because different questions need different modeling answers. And somehow you have to account for uncertainty. We've already heard about different ways to account for that. You have the stochastic differential equations, you can do Monte Carlo, so you have to account for some of that. Then you have to select your model, so you pick one, and you have to somehow determine some parameters for that. Then you do some sensitivity analysis, and during that process you can also decide to adapt your model, to reduce your model, for instance. And then finally you have something that you think, okay, now I can go and look at some predictions, or I can test some hypothetical questions in Silico. And again, there you have the uncertainty. You can do it through a stochastic way, or you can do it through Monte Carlo analysis. So it's a bit of a cycle that you make there. So what I will be showing today is an overview of the different types of modeling technologies that we use in the lab. So I will not go into detail of the equations. I'm happy to talk about the equations with anybody who wants to, or refer to the papers that we've published on that. But I just want to show the overview. And not for every example, I will very strictly follow this loop and show you all the details. But I've tried to put these things together so that you get some of these elements for at least one of the examples. And the last one is the type of models situated in a slightly different way, starting from black box models, where you start only from the data, do machine learning, gray box models, where you try to insert some of the mechanistic information that you have, to white box models, which actually start from the first principles. And when we have white box models, sometimes we actually make the move back, and we make surrogate models, because, well, it's nice to have a white box model, but it takes forever to calculate. So you make a simplified version of that, and then you're back at something that is feasible to calculate and do optimization on. So the way this is structured is 4 groups of examples discussing the cells, the carriers, the culture, and then the in vivo examples. So starting with the cells, one of the questions that we have is, well, we have this cell bank. We have from the orthopedic surgeons, we get periosteal cells, and then we have to culture them. But for now, it's very artisanal. You put it in a plate, and then you put it in a tea flask, and then every week you go and check whether it's almost confluent, and then you do the passaging. And if it's not confluent yet or near confluent yet, you give it another 3 days or 5 days. So it's a bit, it's not very well standardized, let's say. We know that these periosteal cells that has been proven in the lab before that they have stem cell capacity, and we have the luxury of having now 450 donors already in the bank. So different ages, different genetic diseases. So we thought, okay, well, maybe we should just check what is the information that we have now in all the data sheets that we have collected. Can we come up with something to help answer the question, well, how can I predict when I should passage my cells instead of just go and see and then wait for a few more days? So we started looking at the data set that we had. You see here typical growth curves where you have the different passages. A passage roughly is 2.5 population doublings. So you see here cumulative population doublings over time, and you see that we can go quite high with the periosteal cells that we have, almost up to 20, 30 before they go into senescence. If you'd separate them according to age, you see what you expect to see, which is the young cells, they grow faster. The older, that's the top layer, the older cells grow less fast. So those are kind of the things that everybody has seen before. And then we started to use this data, the information that we had on the amount of cells that were seeded, the surface we had, the age of the patient and the gender of the patient, and see if with that information, because that's all we had, if we could have some prediction. So we did random forest, and we came up with this predictor of time to passage. And what you see here are the different passages. Here you see the mean prediction error. So that means that we predicted it will take 7 days before we have to subculture our cells. And this is the deviation in days with respect to that. Now we tested different models, so I would like you to look at the orange and the yellow. These are based on the historic data and on the theoretical calculation. So that is, let's say, we check at 7 days, and you see that there's quite a big, well, we're quite off from the 7 days. And then with the random forest, these are with the random forest, we can get much closer to a good prediction of when we need to subculture the cells, and that throughout the different passages using the information that you have from the cells. So this is a very simple example that leads to a practical application in the lab where people now have a better handle on when to subculture cells. And people have taken this further in the lab into a startup, MyCellHub, where they're actually going to use that also together with the data management system to help the culture, to help raise the quality of cell culture in the lab and in other companies. Now, another question with cells, of course, is when you have these cells and you want to differentiate them, well, what is important? We know that stem cells can be regarded as being on the top of this mountain and then roll down into valleys when you give them certain cues. But where is my cell when I implant them? Where is it in this landscape? Is it in a stable state? Is it in an unstable state? And how do I get it in the state that I want? So that was another set of questions that we were looking at, and we were looking at that from the point of view of the endochondral ossification tract. So the example there that we want to mimic is actually nature's way of making most bone, which is in the growth plate. So we want to look at the growth plate and see what are the main drivers of cell fate in the growth plate. So based on the literature, 80% mouse-derived, based on the literature, we came up with this network where you have the main pathways of the endochondral ossification, of the growth plate chondrogenic differentiation. So you have the growth factors, you have the transcription factors, and then you have all these interactions, the activations and the inhibitions. So this is a static picture. Now, depending on the exact question, you can use different modeling formalisms to make this dynamic, going from the simple Boolean where everything is on or off to the much more complex ODEs where you need so many parameters that we've only done this for instance, two very specific, the BMP and the Wnt pathway, and looking at their interaction, because otherwise it becomes untractable. But the thing that we actually developed was this additive model to have some kind of in-between version where you can actually make a distinction between the protein level and the gene level. So for each, so you actually have two sets of this network, one which are the protein-protein interaction, and then there is the gene level, so there's an interaction from the protein level to the gene level. So you have these fast interactions, and you have the slower interactions. And also every box that you see can have a value between 0 and 1, which means that we are no longer in this on-off situation, and that you can also look at dose response. So the first thing we did is to see whether with this model we were able to capture the major stable states in the growth plate. And so since this is just a cell-based, one intracellular model of a single cell, you cannot have the spatial degradation or the spatial difference. So what we did is we mimicked the inputs coming from the perichondrium, and that actually was the input that drove then the network model. And you see that in pinkish, or this light color, you have the information from the literature, and black is what we had with the multi-level Boolean model. And you see that nicely for the different zones, we capture the on-off information and even some of the quantitative information of the different pathways. So when you have that kind of model, then what did we do with that? Since this is one cell and all the cells are slightly different in the way they are initiated or the levels of the different genes and proteins, we did a Monte Carlo approach. So you take 10,000 simulations where you start with a random initialization of this network and see where it leads to. So this type of cell with this type of intracellular network, is it most likely to go to a hypertrophic chondrocyte, which we call the RUNX2 state, or is it most likely to go to a SOX9 positive state, which is the proliferative chondrocyte? So that is something that you can do in a canalization analysis where you just let it run. Then the second thing that you can do once you have a model where you know, okay, now I'm in the hypertrophic state, or I'm in the proliferative state, how likely am I to change to another state given certain perturbations? So we did a perturbation analysis where you do knockouts or overexpressions of the different boxes, and then you see what is the effect. Will it go from a SOX9 to a RUNX2 state or not, and will it stay there? So you can actually do this large-scale screening experiments that are very costly if you want to do them in the lab, because all these components, they cost a lot of money, and it will take a lot of time. And here you can see, so we did an upregulation of all of these factors and a downregulation. And what you see here is the effect of the stable state. So for instance, if we take FGFR3, when you upregulate that, you drive actually the cells towards hypertrophy at the expense of the cells in the proliferative state. But when you take it away from the medium, it does not have an effect. So it helps to push the cells towards hypertrophy, but if you want to keep the cells in hypertrophy, you will need to add something else to the medium. Now, this exercise was repeated, we made some predictions for different experiments that we did in the lab, different pilot experiments, where we looked at iPS cells, at periosteal cells, and at a mouse cell line. And we were always able to somehow predict which was the best combination to use for that particular example. What we're doing at the moment is, instead of changing one, doing this agonist-antagonist simulations where you try to attack different processes from two sides, from a positive side and a negative side. But then how you validate something like that? Because we made the model because you couldn't just think about a network and try to reason your way through it. So when you make a network model, you cannot reason your way through it. And the fact that we made this additive model meant that we couldn't use any of the tools that were developed for typical networks. So we started from the data again. So we did an inference exercise where we looked at all the mouse-based microarray information that we could find for the growth plate. And since we weren't bioinformatics experts, we just used 9 different inference methods. And then we made a consensus network. So if there's a link that was predicted by all 9 methods, that's the link that we took in the inferred network as well, which doesn't give you the best network, but it gives you the least bad network. And then the way you use that information is then to see how is the correlation, or how good is your literature model as a predictor for the data-derived model. And here you see a typical receiver-operator curve, which kind of tells, well, if this line would be here at the diagonal, it would be just a random prediction. I mean, if you would pick a random network, it would do just as well. The fact that it's over means that it's more than a coincidence that there is an agreement between your networks. Just to have another validation step, we also made a string network. You can go to this database, and you can say, build me a network with these pathways, so many nodes, so many interactions, and it will do that based on the information that is available in the literature. When we analyzed that network compared to the microarray inferred network, you saw that there was a very poor correlation. And the reason for that is that most of the string data is derived from cancer or maybe cardiovascular, but very, very little of it comes from actually cartilage and bone. And so those kind of tools we have to use very carefully. So the old-fashioned labor-intensive go to the literature and make it yourself is still very useful. So this is what we did with the cells. If we then go to towards the carriers, we go from the chondrogenic differentiation more to the intramembranous ossification, where we use calcium phosphate-based carriers. And the questions there are what carrier to pick. You have 20, 30, 40, 50 different types of commercially available, clinically used calcium phosphate-based biomaterials. Now, how is the biology influenced, and which carrier should you pick for the cells that you have? What will happen in vivo with that? So there was a lot of experiments already going in the lab, so we took a more reverse engineering approach there, where you can look at the scaffolds, derive some characteristics, look at what bone formation they give, get some insight on what is important or not, and then use that to build your own scaffolds. But for now, I will go up till there because the building, that's still ongoing. So what we had is we took the carriers, we analyzed them, look at their chemical composition, at their structural composition, also using nano-CT to have a 3D insight, combining that with the cells, again, the periosteal cells that I mentioned, and then implanting them, since it were human cells, in nude mice, and then taking it out at certain stages to get the information. And again, using nano-CT to analyze it. For instance, what you see here is what you can have with contrast-enhanced nano-CT compared to a whole mount staining, where you nicely see the difference between the bone and the cartilage because we were using hexabricks as a contrast agent. Now we have developed new contrast agents that are less detrimental for the soft tissues, that can actually allow histology to take place afterwards, and that allow us to also look at the angiogenesis and the bone marrow formation, for instance. So we have then this whole bunch of information. We have the pre-implantation information of the scaffolds. We have the information with 3 days after implantation. We looked at some of the biology, so the proteins, the gene expression. 12 days after implantation, we did the same thing. And then 8 weeks after implantation, you see bone formation. So there we quantified bone formation. And for instance, what you see here is a cross-section of a 3 millimeter by 3 millimeter cylinder that we implanted in the mice. And you see this is still a calcium phosphate grain, and this is bone that is being formed. And you can see these are 5 examples of the biomaterials that we tested, that they're very different. Even though they're all calcium phosphate collagen based, they're very different in structure, in morphology, and also in bone formation. So if you do a very simple, again, partially square regression, because it's using only the data, and some of the biological colleagues prefer to use only the data. Well, what you see is that, for instance, if you use all the information that you have available at day 3, you can have a quite good prediction for the set, the set that we were testing, as you get some kind of linear equation, which has some of the biology in there, some of the protein expression, and some of the biomaterial characteristics. Now, of course, nature is not that simple, so we took another sample set, and we tested them. And then it performed relatively okay, except for this one. And this one, reprobone, is from this whole sample the only one that is not a calcium phosphate collagen combination, but a pure HA one. And I was very happy because that proves the point that if you take something that is so simple that you can only use it on a data set that is really restricted. So that was a good message to pass on. So if you want to have some more mechanistic insight, you have to use a bit more complicated models. So what we did there was look at 2 things. Well, first you need to understand what is the amount of calcium, for instance, that is being released from the scaffold, and then you can try to simulate how the biology then responds to this environment. So what we did is we made a model where we took all these grains, so it's based on the CT. We defined the geometry, and so and then we use the level sets to simulate the degradation. So basically, you take the interface and you move the interface according to a certain law or a certain process that you define. And then, so we could simulate both, if I find my mouse, you can simulate both the degradation and the release of the calcium at the same time. So in the color is the calcium concentration, local calcium concentration, and you see that the grains are dissociating completely in the end. Now, trying to capture that in very simple degradation laws was not so easy, because either we were able to fit more or less the beginning and then we didn't match to hit the end, or we missed the beginning and we're closer in the end. So there it seems that it's not just one set of dissociation constants to use, but there's now been people reporting in the literature that you should actually use 2 constants for the different phases. So that is what we're looking at right now. So if you do things like that, you can have a degradation, you can simulate, and this is the real implantation degradation. It doesn't go away completely, so it's much more subtle. But you have some idea of the local calcium concentration. You can couple it to an equation or a system of equation describing what happens to the biology. So for instance, this one was established together with the experimental collaborators, just focusing on the HPDCs, the stem cells, turning into osteoblasts, so the direct bone formation, making osteoid, making mineralized tissue, and the growth factors that were involved. And again, that was first implemented in MATLAB, but then recently also in FreeFem to combine it with the level set. So just as an example, because this is still in process of being calibrated with the experimental data, I would like to show 3 cases where you have different combinations of growth factors and biomaterial release for the same geometry. And you see, after you run the mathematical model, what you see here is the bone formation after 8 weeks. So you have bone either right next to the grains in red, you have bone in the open spaces between the grains, or you have bone just outside of the implant. And the reason why I want to show you is because it corresponds to the 3 different types of bone formation. So I'm not saying this is the perfect model for that type, but I'm saying is that with this type of equations, with this type of technology, you can capture the different types of bone formation. So if we somehow are able to calibrate, we're fairly confident that we will be able to capture the different experimental things. What you see here is the bone formation close to the grains in the open spaces, and this is typical BMP-driven bone formation where you get an ossicle forming with only bone on the outside and nothing on the inside. So the reason we want to do this is then to match this with some properties of the cells that we have. For instance, this is just a theoretical projection where you take different amounts of stem cells that you can start with. So in the black is when you have a very low percentage of stem cells in your cell pool, in orange when it's very high. And here you see different scaffolds just brought down to their calcium release property. And if you have only very few cells, well, you should be very picky which type of scaffold that you use. If you have a lot of cells available, well, then you can do some optimization, but basically most of the scaffolds are fine to use. So that is the way in which we would like to use this in the end. This is something that you can not only do with calcium phosphate-based biomaterials, but we're also doing for magnesium-based, so biodegradable metals, where you can use the same modeling technology, where the question is the same. What is the best design so that I get a certain kind of concentration of released products, but also a certain type of bone formation? So when we then pass on from the carriers to the culture, to the bioreactor, so instead of keeping our cells growing in flasks, we want to provide them with a 3D environment to be able to upscale. The question there, of course, is how do I control such a process? We were using titanium-based biomaterials because we can 3D print them in any shape and any geometry that we want. But that also means that you can just open it up, put it under a microscope, and look what happens on the inside of the scaffold. So how can I we have sensors here at the outlet and at the inlet. Flow is going like this. But how can I deduce from those types of information, how can I deduce what is happening inside of the scaffold? So there again, we started with the biological observation that cells like to grow in these corners. So we started with a level set method again, where we simulated this interface, and this interface between the new tissue that was formed by the cells, and the void space. That interface was being advected based on certain quantities. And the first thing we did was do it based on curvature. So you have the new tissue here in green, you have the interface in red, and it's being advected based on the curvature. So where the curvature is highest, you have the highest speed with which the interface is moving. Now, the first thing we did was try to see whether it was able to capture some of the static experiments that we had done. So we had 6 different scaffolds with 3 different unit cells, with a hexagonal, a square, and a triangular structure. And you see here the live dead staining. So we took, for the hexagonal, after 7 days, we calculated the filling. We calculated the filling throughout the simulation, and we used the 7-day time point to calibrate the computational time with the physical time. And then all the others were using the same calibration key to come up with this physical time, the simulation of physical time. And you see that we were able to capture the behavior. Well, you see it already here in the pictures, but also quantitatively we were able to capture the behavior, at least at the surface of the scaffold. So this curvature-based growth, when there is enough nutrients, seems to be something that is really universal also for our cells and our setup. Now, of course, if you do that at a 3D structure, the inside will die because of lack of nutrients. So we had to model also the flow throughout the whole scaffold. And we felt it was really important to not ignore the new tissue that was being produced and the flow that was pushed through the new tissue as well. So we used the Brinkman equation that allowed us to alter between the Stokes equation in the free zone and the Darcy equation here in the porous area. So then we had our speed with which the interface was advecting, not only based on the curvature, but also based on the shear stress that we could calculate from the flow, and the level of oxygen that we also added to the equations that was just being advected with the flow. And this was based on the literature, where you see the shear stress, for instance, has some stimulatory effect, but when there's too much shear stress, obviously your cells are being detached, and so the growth stops. And oxygen, basically, this was a very simple curve saying you need some degree of oxygen, because otherwise your cells are going to die too. So just as an illustration, we looked at a diamond-shaped scaffold inside a bioreactor chamber. And I'll show you 2 cases. One where we put the scaffold at the bottom, so at the inlet of the chamber. And you see that here in pink, the filling of the scaffold is not going very well right at the inlet, of course, because the flow is too high and is not well developed, which is also reflected in the poorer filling at the bottom of the scaffold compared to what happens at the top of the scaffold. But if you put your scaffold in the central part, where the flow is nicely well developed, you see that you get a much more homogeneous filling throughout your whole scaffold, which is also, again, reflected in the experiments, where you see a much more homogeneous filling throughout the entire scaffold. Now, the last thing we did, the most recent addition here, is we added glucose and lactate to the equations, not because we like to add these things, adding more parameters to it, but because glucose and lactate are 2 parameters that are really often used as indicators of quality of the biological activity of your process. So that is why we added them anyway, and we designed some specific experiments to measure these quantities to be able to validate that experiment. Now, just to come back to the questions that I asked, can we now do something with the questions with respect to quality control for the bioreactor? For instance, one of these things that people want to measure, or think that people measure in a bioreactor, is the pressure drop over the scaffold. And they want to use that as some kind of measure of filling of the scaffold. What you see here for a gyroid scaffold, for instance, is the pressure drop that you see that rises after day 12. But if you look at the same day in the filling, you see that you're almost at 90% filling of the scaffold. So it's not sensitive enough to pick up the early phases of the filling. Furthermore, it doesn't give you any insight into what happens at the microscopic or at the cell microenvironment level. So using this model, you can at every location, at every point in time, look at what happens in terms of the niches, in terms of glucose, lactate, oxygen, and you can understand the heterogeneity of the environment that the cells in your culture system are confronted with. So if the end goal is to take out the cells after the culture period and to treat them, well, you can see that they have been subjected to different microenvironments, so they might have changed their behavior, and the heterogeneity of your cell pool might have increased. You can quantify this in terms of other things for different geometries. You can play around, and you can see that your glucose, pH, lactate, your shear stress, and anything you want is changing over time and changing for the different geometries that you use. So you can pick your scaffold geometry based on specific requirements that you have for the environment that you want to impose on your cells. Same thing if your end goal is, for instance, I want to get as rapid as possible filling of the scaffold with new tissue. You can do a simulation and see what the effect is of different geometries that you pick and see how fast they are filling or not. One possible goal could also be what is the most optimal culture regime, because we're changing medium every 3 days, but we know in the beginning that's not necessary because, well, there are not so many cells, they're not consuming so much, so we might wait a little longer, whereas at the end we might need to replace a bit faster. So here we calculated, we let the model actually determine the replacement of the medium when we saw that the pH of 20% of the volume was actually dropping below a certain threshold. And you see indeed that in the beginning the blue can wait a little longer than the orange, but at the end you have to replace it much more, or you have to replace it more frequently because the lactate concentrations are getting higher as you have more cells. Now these simulations, they take 3 days to run, which is not something that is really, and it's parallelized and everything, so it's not really feasible to run this for a wide-scale optimizations routine. So we made a reduced version, a homogenized version, where we removed basically all the spatial components, but we kept the same biological mechanism, so the Michaelis-Menten with respect to the nutrients, they were all still in the model. And then you can do a much faster first step in the optimization protocol, where you can do a single optimization looking at what is the best refreshment time, refreshment frequency, but also take into account the cost. Because of course, the more you refresh, the better for your cells, but the higher the cost. So you can do this multi-objective optimization as well. Now that was kind of a zoom out where you let go of the spatial components. You can also do a zoom in, and that is where we actually worked together with some of the people, or we used some of the work that Hans van Oostenrijk presented yesterday or 2 days ago, where we used his agent, where we used the CFD simulations that we have combined with the agent-based model or the individual cell-based model that they developed to try and look at the effect of this flow on the different cells. And we modeled cells, in groups or cells attached to a strut in different ways with respect to the flow. And what you then get, for instance, here at the right, you get the average shear stress on a cell, the maximum shear stress on the cell, and in the white, you have what is calculated just based from the CFD. So you see it's the same ballpark, but you can get some refinement by using a zoomed-in version of the model. So that was for the culture. If we then go to the last set of examples, which is the in vivo, where you start from a fracture healing, which goes wrong in certain cases. So the questions there are, can we simulate these adverse fracture healing situations, and then can we do something with that in the model? This started already quite a while back, where we had a PDE-based model of the different cells, tissues, and growth factors that are important for fracture healing, and we added onto that. So this is the basic model that was developed a long time ago, and we added onto that an individual blood vessel simulation that was borrowed. The description was borrowed from tumor angiogenesis, where you can simulate the different blood vessels inside the fracture healing and its effect on the rest of the development. So we looked at a number of different cases of adverse fracture healing related to mechanics, related to large soft tissue damage. The two that I would like to briefly discuss are the critical size defect and then the congenital defect one. The critical size defect, well, you see on the left-hand side the blood vessel formation, which comes in bumps because we only show the blood vessel once it connects to the existing vasculature, so you don't see it grow, you only see it when it's connected. And you see here the bone formation in black, the intramembranous ossification, and now once you have the vasculature, you also see the endochondral ossification taking place. On the right-hand side, you see a large defect, large for in dimensions for the rat or for the mice, where after 90 days, basically the whole process has stopped. It's not because we stopped the simulation; it has stopped. And what you see is that you get some bone formation that is closing off the end of the fracture on both sides, and nothing happens in between, which is similar to what you see in the experiments, albeit that we still have a bit too much bone formation taking place here. So what you can then do is look at your model and see what is the critical factor that made your biology stop. Well, and no surprise, it turned out to be the oxygen that went too low for the cells to be able to survive, even the chondrocytes. And by the time the vasculature, which grows at 35 micron a day, had restored a little bit, the cells were gone. And so there were no more hypoxia signals to attract the vasculature even further. So the whole thing stopped. One of the postdocs in the lab was then working on the production of oxygen-generating beads. So they would be able to produce oxygen for a certain amount of time. They could be injected into the fracture site if you had a large fracture. And with the model, we made some basic predictions on how many beads do you need and how much oxygen do they have to release for how long to be able to sustain the biology, the native biology that is available there. And this is an in vitro experiment that we simulated where we had just a hydrogel without beads and with a certain level of beads. And basically what the beads do is they keep the cells alive in such a way that they're not too comfortable, that they feel they have enough oxygen and don't do anything. They are alive, but they're still they're still a bit oxygen deprived, so they're emitting all kinds of VEGF and other angiogenic factors to attract blood vessels. And the first histology results, and I appreciate that this is a very selective shot, but this is what we have for the moment, where you see that in the case of the particles that were used, you get a much more healthy blood vessel formation that took place in the hydrogel using the beads. So the final example, and with that I would like to go back to the very first thing I said, which is the non-union, the NF1-based non-union. Actually, what we know of neurofibromatosis is that it affects certain processes of the fracture healing, and those are actually variables in the model that we have. But we know that there is a very wide variety in phenotypes of neurofibromatosis, and it's not necessarily linked to the exact location of the defect in the gene. It's really hard to do comprehensive studies because you have 500 patients in Europe. Many of them are not specifically treated or are not diagnosed with NF1. They just have a non-union fracture. So it's really hard to do all these comprehensive studies if you have only case reports. So instead of going for one NF1 type non-union, we decided to take 200 combination of these 8 parameters that were affected and change them between severely affected and not affected at all. So you had 200 digital phenotypes, if you wish, of the neurofibromatosis case. And then what you do see is, if you look, this is just 1/4 of the whole callus. What we did see in a large number of them, the typical characteristics of an NF1 fracture, which is a lot of fibrous tissue, some cartilage, but quite late, and then no bone formation at all. But there is vascularization starting up. So these are the typical characteristics of an NF1 fracture. So if you have these 200 patients, the thing that you can do, which you can never do in a case report, is give them no treatment and give them a treatment at the same, or in the same patient, test different strategies. So what you see here in the graph are the 200 dots, the 200 digital phenotypes, if you want, where on this case we have the degree of non-union when there was no treatment given. So these are the ones that are healing. These are the ones on the right-hand side that are not healing if you're not treating. On the vertical axis, you give a BMP treatment at the moment of fracture. And so the lowest part, these are the ones that are healing, and these are the ones that are not healing. So the green ones, they heal with and without treatment, and we do see that there's a number of NF1 fractures that do heal, but you can appreciate that there is quite high values for this complexity index, meaning that there's quite a high degree of fibrous tissue in the bone, so making it very likely to refracture. The black ones are the ones that are responding to treatment, so they weren't healing, but they heal when you give them BMP. The blue ones are the ones that are non-responsive, and again, also there we have case reports to substantiate this. And then the red one, well, before I go there, basically with these three cases, you could say you give them BMP, whether they heal or not. And then we had this fourth category, which actually healed without the treatment, but if you gave them BMP, it interfered in such a way that the healing stopped prematurely. So that was actually adverse response to the treatment. So now we need to increase the number to see if this is just a numerical fluke or where this exactly is coming from. And what we then did is use this 200 phenotypes as actual patient and tried to see, okay, what in these model parameters, which are the things that we can measure of these models, what could we use as a biomarker to put them in the different categories? And so it turned out to be cartilage formation that was able to clearly distinguish between the healers, the responders, and the non-responders, but not the adverse responders. Well, the group was too small anyway, but here you saw a nice division based on this information in the cartilage formation. What we had, we have also a few cells, or for a few donors, we also have the periosteal cells, so we did the trilineage differentiation test, and you see that in the degree of chondrogenic differentiation and cartilage matrix formation, there is a variety between the patients that are really severely affected that do not heal and the patients that responded to treatment. So this is kind of what we're trying to further develop now to see if we can get some more correlation between this very basic test that we can do on the cells of the patient and know a little bit beforehand already in how much danger they are of not healing even after treatment. So these are the different examples that we had. And I would like to end, or I had already foreseen it, it's a bit going in the same direction as some of the last things that Alfio said, is how do you establish credibility for all these things? Because we can make models all we want, we can publish in our journals, that's all fine and dandy, but you want it to either go to regulatory, you want it to go to the clinic, so how do we do that as a community to establish this credibility? So the first thing is that you need to be able to report. So if you go to a regulatory office and you have a submission, a file, you need to report your models in a certain way. So FDA has produced these guidelines. If you have a finite volumes model, you need to show them this and this and this information. If you have a finite elements model, they expect this kind of information. So that is what they expect. Then the second stage that they're working on is the VVUQ, the verification, validation, uncertainty quantification. Well, verification is very clear that you didn't make any implementation mistakes. The validation is that, of course, your model corresponds to physical reality. And then the uncertainty quantification is how does the uncertainty on the assumptions that you made, on the parameters that you use, on the measurements that you used, how does the uncertainty on those things affect your solution? So what is the context of use of your model? What is the context in which the prediction of your model can be trusted? And that is something, especially the VMV part, is now being worked out. There has been this exercise by the medical device consortium, the ASME and FDA, to come up with a standard, which is called the VMV40. They have VMV10, VMV70 for automotive things, for airspace things. Now they have one for medical devices where they say, okay, if you want us to trust your model results as part of your submission, this is how you have to show the verification and validation. And they're working on getting some ideas on how to do the uncertainty quantification. So these efforts are not just the FDA, these efforts are a consortium of people that are doing this. We're trying to transplant this also to the European setting. And we, in this case, is the people at the FDA. It's also the VPH Institute, so all the academics that are there, and our partners in industry in the Avicenna Alliance. And for that, we also need policymakers. We need to push from the politics and the policy to help move things along. And one other thing on efforts that are ongoing to make sure that we're all talking the same language. I just got here from a workshop at the Geris, the Joint Research Centre of the European Commission, where they were working on a project called Bridging Across Methods in Biosciences, where they want people from in vitro and silico and in vivo to talk to each other, to kind of find a common language so that we can use good quality in vitro data, good quality in silico models, and that when we say we validated our method, that we know what the other person is actually talking about. So these efforts are ongoing, and I think that as a new generation of scientists, that we should actually be aware that we need to do these things. When not publishing our model is not good enough. If you want to help the patient, these are also the things that you need to work on to establish this credibility. But the rewards might potentially be very high, and this is one of my favorite slides also from this interaction between the Medical Device Consortium and FDA. This is what they project as the future for information in clinical, in dossiers that get submitted to the regulatory office. Today, 1/3 of the information comes from in vitro tests, 1/3 from animal tests, and 1/3 from clinical data. Very little information is coming from computer models. They project that in the future, almost half of that information will be replaced in some shape or form by in silico information, be it replacing the animal tests or the laboratory in vitro tests, or doing in silico clinical trials before going to the actual in vivo patient trials. So I think it's looking good, but it's up to us now to make sure that we have the models that can be trustworthily used in this setting. And of course, I did almost none of the research that I presented. It's the people in the lab that have actually done all the work. It's the funding sources that allowed us to get all the people and to gather all this data. So I would really like to thank them. And then I would like to reiterate the invitation of Esther for the VPH conference in Zaragoza. But if you happen to be in Brussels the day before, there's also going to be an event in the Parliament organized together with the industry partners of the Avicenna Alliance, where we're going to try and convince policymakers and people, well, people in places with power, that this is something that is a very important part of the future of healthcare. So I would like to invite you to all of those things, and thank you for your attention.
Well, Liz, thank you very much for this wonderful journey through translational research, tissue engineering. I think it puts a lot of pressure on many of us. So, questions?
Hi, Liesbet. Very nice talk. I had a question with regards to the part where you did the modeling of your different scaffolds in vivo. Which features, when you were modeling that to say which of these were most osteoinductive, which features of the scaffold were taken into account, or which one of those kind of arose as being the most important elements or characteristics of those scaffolds?
For now, we use conditions that are not detrimental from the point of view of oxygen or glucose or lactate, because we simulate the 3-day, every 3 days having a replacement. So it's the curvature and the shear stress at this point in the model that are most important. But that is something we've validated we've calibrated that, let's say, for the gyroid model. And the D-cup, so those are triple periodic surfaces where you have the geometry described by combinations of sinuses and cosinuses. So curvature is an inherent part of that. But we have to now redo the exercise for the more classical diamond-shaped or triangular-shaped ones. So there possibly we'll have to see if that balance that the two most important ones, being curvature and shear stress, are also still most important for those classes of scaffolds. That's what we're working on right now.
And for the studies that you did in vivo, were those done orthotopically or ectopically?
The fracture healing ones, so the large defects, the soft tissue damage, the mechanical ones, which are literature-based, those are all in vivo or orthotopic, because the fractures. The one that I showed with the oxygen generating beads, that's an ectopic one. And the other one, so with the biomaterial, those are all ectopic. For the ones that are most promising, the most promising combinations have then been taken orthotopic, because orthotopic in mice is so much more cumbersome that we first do ectopic screening, even though we're not entirely 100% certain that ectopic is always the best predictor of the orthotopic one.
And then the last question. For those, again, like the BIOS, the NUOS, et cetera, that you had done, so you talked about like calcium release and so forth. Are those models also able to take into account like mechanics, the effect of mechanics?
Not at this point. The reason that mechanics is not present in most of the applications is that we work for a clinical application that has an external fixation, that has the lizard cage, which we have for the mice, the rabbit, and the sheep. So we assume that the mechanics is brought down to a level where it's not playing the major role in the process. But of course, for ectopic, this is on the back of the mice. They roll onto it, so there is mechanics for sure. So this is something that we have ignored.
Okay, thank you for your talk. It's incredible. I just have a one short question, but I would like to know if you are planning to include the roughness of the surface in your model.
That's actually one of the things. So, as I said, we stopped at the phase where we now need to do the reverse engineering. So, Bing, who's here, is actually working on the design of scaffolds that will be 3D printed in calcium phosphate. So, that's something that we're working on, and there the roughness of the post, let's say, post-printing treatment is going to play a role there. There has been more than enough evidence in the literature that that's important.
Any other question? Well, indeed, I have one. It's more philosophical than scientific per se. All what you've shown seems to be the result of an extremely mature view towards a specific objective to treat a disease, and the whole pipeline of methodology as it is required for this. So it contrasts with several talks that we had that have been mostly driven by methodology. So people wanted to have elegant methodologies as precise as possible, but then applicability is not straightforward, at least yet. So the question is, along all these years of research, more than 10 years probably, so has that always been the vision, or very some aspect has been first methodologically motivated, and then you have found an application and you have refined the overall vision?
It's two aspects. So one, I was really lucky to have the Prometheus environment where Hans was in there before me, so there was this concept of modeling is important, and sometimes we need to do an experiment that will not lead to a Nature publication, but just will be used as an input for the model. So that environment was there, and that's really important because that generated the questions. The reason why this is not methodology based, and that's maybe that's just a clever way of selling the fact that I am not very particularly gifted in any one methodology. I'm just very curious about different things, and I maybe don't have the patience to dig so deep as, for instance, Alfio has in all of the equations. So I'd like to use methodologies that are mature enough, that have been prepared by other people, and see if we can use them to answer the questions that we have. So it's a combination of just being curious into different aspects and then selling it in a way that it sounds like this was all, a big master plan from the start. But it's a combination of different factors.
My question was not about the fact that it's not methodological.
No, but so it wasn't a master plan to start this way and to not focus on a methodology. It was just from wanting to test different things instead of digging deep into any one methodology.
Yes, it has been more vision driven.
Yeah.
Any more question? No? So we're on time, so it's time for lunch. So thank you again, Lies.